So our first exam question states, describe how you would use cell fractionation and ultracentrifugation to obtain a sample of mitochondria from muscle tissue. Okay, so what you're going to do is you are going to first of all homogenize. So the first step is you're going to homogenize. And how are you going to do this? You're basically going to you're going to cut open the muscle tissue. You're gonna once you've cut open the muscle tissue, you're gonna add it to a cold isotonic and buffered solution. So add this to a cold isotonic and buffered solution. And the reason why this is, it's only 4 marks, but I'm going to briefly put the reason down. It won't be fully required for a question like this, but I'm going to add the reason in. To prevent damage to the organelles. Okay, and then what you're going to do is you're going to blend it. So you're going to blend this solution and you're going to filter. And we're filtering to remove large debris. Okay, once we've done that, comes our centrifuge. What we're going to do is we are going to centrifuge at a low speed, and then we're going to remove the supernatant. Okay, so we're collecting the mitochondria. The mitochondria is the third densest organelle. So what we're going to do is we're going to centrifuge initially at a lower speed to remove the nuclei. And because this is not a plant tissue, there are going to be no chloroplasts involved at all. So we can just skip the chloroplast because it's not going to be there in the solution. So we're going to centrifuge at a lower speed to separate out the nuclei, to remove the nuclei. And then we're going to centrifuge at a higher speed to collect the mitochondria. Okay, so here's the mock sim. You use a cold buffered solution with the same water potential, except isotonic for same water potential, which you can see. Homogenize tissue and then filter. It says accept blend or grind. For homogenize, you centrifuge at a lower speed to remove the supernatant or to separate out the nuclei, or you could have said in terms of density there. And then you centrifuge the supernatant at a higher speed so you can collect the mitochondria. So that's where the 4 marks are coming from in this question. And this was from 1 year's paper. I've got another question which is literally very similar. Okay, and this is from a different year's paper. It says describe and explain. So this was just describe how you would. So you didn't need to go into sort of any detail about preventing damage to the organelles or to remove large debris or anything. You didn't need to say that because you just said describe. This one is 6 marks and it says describe and explain how you would use cell fractionation and ultracentrifugation to obtain a sample of nuclei from muscle tissue. So I'm just going to show you the mark scheme for this question. So again, so it says describe and explain. So each step that you do, you also need to explain a reasoning behind it. Okay, you also need to explain why you're doing this. So we know that the first step is homogenize. You won't get the mark if you don't explain why you need to homogenize. You homogenize to break open cells. Okay, you then filter to remove the debris. Okay, you need to put it into a cold buffered isotonic solution. Why does it need to be that way? Is so you need to explain it. Every reasoning that you give, you need to explain. So it needs to be cold to prevent enzyme activity and prevent damage to the organelles. It needs to be isotonic to prevent osmosis or prevent bursting or shriveling or anything like that. It needs to be buffered to stop enzymes or proteins from denaturing or to prevent damage to the organelles. Okay, and then what you do, you centrifuge at a low speed. So the nuclei, this is the densest. We're removing the nuclei. So the nuclei moves to the bottom. Okay, and that's your 6 marks. Okay, because it says we need to obtain a sample of nuclei from muscle tissue once again. Okay, so as you can see, these mark schemes, this is just the describe mark scheme. Okay, so if you get a question on describe, you just need to outline the stages, the steps that you would go through to centrifuge a sample. If it says describe and explain, you need to describe the stages that you would go through, and you would need to explain why you're going through each of the stages. So over here, homogenize to break open cells, filter to remove the debris. It needs to be cold to prevent enzyme activity. It needs to be isotonic to prevent osmosis. It needs to be buffered to stop enzyme activity. And then you centrifuge at a lower speed so you can get out the nuclei, which moves to the bottom as it's the densest organelle out of the whole cell.